Maria Garcia Alai. Can you hear us in the lecture theatre for questions later?
I can hear you. Can you hear me?
Yes, thanks. So
Great.
you can go ahead and present. Thanks very much.
Thank you. So thanks a lot for the organizers for the invitation. And yeah, unfortunately, I cannot be sitting there with you. But nevertheless, I'm happy to present my data about how to prepare good samples. Oh, sorry, it looks like it doesn't go to the next one. Yeah, good. So it doesn't matter how fancy equipment you use if you are doing structural characterization or biophysical characterization of your sample. If you start from a bad sample, you end up wasting a lot of time and a lot of resources, and you will end up with, of course, bad results. So there are quite some things that we can do for preparing good samples, and we don't necessarily be relying on learning from our own bad experience. We can do some check quality control before starting. And there are three main things that cannot be forgotten. The fact that you work with purity higher than 95% if you're willing to do structural studies. Homogeneity, so to avoid aggregation, and there are different biophysical techniques like size exclusion chromatography or dynamic light scattering and combinations of those to check that you are having actually a monodispersed sample before you produce crystals, for example. And very important is to identify that you are working with the right protein and not purifying something else that comes from the host organism, and that your protein is the integrity of your protein, that you are not lacking, for example, some amino acids at the N terminus or C terminus, or you have some degradation. And then there are other things one can check, something as simple as doing a UV spectrum, check batch to batch consistency, because sometimes the quality of the sample changes if you had some problem during purification with one of your purifications. Check the stability and the folding state with techniques like circular dichroism or differential scanning fluorimetry. If you're working with enzymes, it's very important that you check for the amount of functional protein that you have, the active protein, that you have not damaged the functionality of your protein during purification, and the optimization of storage conditions if you're willing to save that for later. And then you can be sure that when you get it out from the freezer, your sample is going to be okay. So our work in the sample preparation and characterization facility is enabling structural studies for biology research. We are sitting in Hamburg in the synchrotron, and we have two big platforms, one for high throughput crystallization and another one for molecular biophysics. And basically what we do is we guide the user that sometimes is an expert, so they know exactly what to do, but we still guide them. Or if they are not so familiar with structural studies, we even help them in the design of the experiments. And all the way through to data analysis, we have a lot of instruments for measuring interactions between biomolecules and doing characterization. So some of the examples of the pipelines that we produce, we try to deliver tools to help the students to prepare good samples. For example, this is an example of a pipeline for preparing membrane proteins before going to cryo-EM grids. And we combine different techniques. We start with a purified protein that has a very nice size exclusion chromatogram. Then we perform differential scanning fluorimetry to find which are the right conditions of the protein buffer and detergents. We also do dynamic light scattering to see if there is any aggregation. And finally, we use mass photometry to see if kind of at the single molecule level, if you are having for example, the presence of the complex that you are looking for. And we produce all these type of protocols or pipelines available for the community. This is an example of buffer optimization. Sometimes you start producing your protein and purifying your protein, especially if it's a membrane protein. something that you have to be careful is when you select the right detergent. One thing one can do is to check for dilutions in different detergents. Here, for example, we were using 96, but you can survive probably with 20 or so. And you can do a simple thermal shift assay if you are using differential scanning fluorimetry that allows you to check for the intrinsic fluorescence of the tryptophan. And you can find out in what condition your protein is more stable. And then you can visualize it kind of in a deep well plot where your protein is let's say happy, green, or aggregated and unfolded, red. The thing is, when you produce this type of curves, and these are unfolding curves following fluorescence flow data, you can get, you start from a folded state, and then you start heating up your sample. Ideally, you get one inflection point that corresponds to the TM, so the protein, the melting temperature of your protein, and then you reach the unfolded state. And sometimes the curves are nice, but when you're working with more complicated samples, membrane proteins is all these cases, you can see that you can get what I call set curves, that they go on the other side, but still in these three examples, you can see that the inflection point is visible and nicely detected. But sometimes samples show this type of profiles where it's very difficult to find the TM. Sometimes this can be because simply the fluorescence signal is not good. The quenching is giving you a flat curve. Or sometimes it could be that your protein is already unfolded. So there are some things one can do to survive this problem. And it could be that also instead of only looking at the fluorescence data, one can look at the aggregation state. So if you are following dynamic light scattering or simple static light scattering, you can get kind of this similar curves. Instead of getting a TM from the fluorescence curve, you can get mid-aggregation point of your sample. Then you can look at the first derivative where you are visualizing that inflection point as a maximum or a minimum, depending if it is an S or a Z curve, as I was showing before. Again, some samples are nice and you get a nice clear peak, and sometimes you get this type of profiles when one has to be more careful. Another thing that it's very important if you are looking at the stabilization of your protein in different conditions, if you are checking for salt concentration in a specific buffer, for example. Sometimes your TM is okay and you are willing to have a high TM for your protein to be considered stable. Let's say something at 40 degrees, it's nice. But one that would be, the TM would be 50% of your protein being folded and 50% of your protein being unfolded already. So you don't want to reach to that state. And then you have to be checking at the onset of unfolding. So at what temperature, let's say something like 2% of your protein will start to be unfolded. And you try to work at temperatures that are lower than that so that you minimize any aggregation. So once you have done all this buffer optimization screening following fluorescence or aggregation, one can visualize the curves. And what we have done in the lab, and I think it's very useful, is we have designed software to help you to analyze your data. So the first module that I'm introducing here, and it's available in a server, is called MoltenProt. And you can upload all the data that you obtain from a thermofluor experiment or from a nano-DSF experiment or from a dynamic light scattering that allows you to do thermal shift, and you can upload this on our server. So here I'm introducing eSPC, that it's an online data analysis platform for molecular biophysics. We have different softwares. The one that I was talking before, I'm just mentioning for buffer optimization. And you can also follow other biophysical techniques like fluorescence, dynamic light scattering, microscale thermophoresis. It's a user-friendly software. It behaves also as a virtual teaching tool. We have lots of YouTube tutorials, and you can even check the quality control of the data. So if you have this type of curves that I was showing, the software will tell you if this is a good curve or a bad curve. It's free for academic users, open access and open code upon request. And you can upload data from different technologies. So this is an example of if you enter the server where you find, for example, tutorials, and you can watch all these videos online that will explain you step by step how to analyze your data. All the modules that are in our web server more or less have the same way of functioning to make it easier for the user to understand the domain and the objective, the data collection and the selection, the pre-processing, the modeling step. For example, for the fitting of the unfolding curves, you can choose different models. The most common one is a two-state model of folded and unfolded state. And then evaluation and interpretation of the data, where you can also check the quality control. These are two new modules that we have just incorporated to our softwares, and is one for mass photometry and another one for dynamic light scattering. So mass photometry is kind of a new technology that allows you to measure in solution and to address the molecular weight of the complex or the protein that you are working with. And you work at very low concentrations, so this is very useful, for example, in the case that you want to prepare samples before going to cryo-EM. You can see if you are dissociating your complex when you are going to the grid. Here we have an example of a complex that we were working in the lab, and actually we could have two types of complex, one that was around 300 and another one that was 400. And we wanted, the one that it's around 300 was missing some of the subunits that we wanted. So we had to try to push the equilibrium to get the right oligomeric state. And this technology has been very useful in that case. You need to calibrate the instrument with different standards. And also in the pipeline that I was showing before, in that publication, we are showing even how to calibrate the instrument. Another thing one can do if you have, for example, two proteins that are interacting, or a protein that it's binding to a small ligand, a compound, or a peptide, one can, of course, do the gold standard and try to measure the interaction by ITC, isothermal titration calorimetry. One can also do MST, microscale thermophoresis, and there is a module for that in the server that I'm presenting. But sometimes the KDs that so the interactions that you're looking at are weak. And you would need so high concentration of the ligand in the syringe, for example, in the case that you're doing ITC, that it would just not be feasible. So here what we are presenting is another method that you can estimate binding affinities by isothermal analysis. And you can do this by titrating your ligand on your protein and running thermal shift assays. So here in this example on the left, what you can see is different curves. And the blue curve corresponds to the unbound state. And then when you start increasing concentrations of your ligand, you see that you get a shift in the TM. So the complex is more thermal stable than the unbound state. And a lot of people try to calculate an apparent binding constant by looking at this thermal shift. What we are suggesting is, because this is not really thermodynamically correct, we are suggesting to stand at a certain temperature, so on the vertical zone now, and calculate the fraction of unfolded and unfolded state as a function of the ligand concentration, and then do the fitting. Of course, you need to be careful at what temperature you choose, And by a rule of thumb, you should work around some degrees shifted for the TM that you obtain for the unbound state. So all these models and the thermodynamic theory can be found also in the server that I was presenting, and can help you to choose which is the right model for your case of study. So here, for example, a protein where we did a kind of a benchmark of this method, isothermal analysis, with the protein that we know we had very well characterized the binding by ITC. That was a kind of tight binding, 200 nanometers at 20 degrees. And then when you stand on the temperatures that are close to the melting point of the unbound state, that would be the blue line here, you can calculate the TM by the isothermal analysis. You see the numbers that you get are not the same. You cannot think of an absolute value, and this is of course related to the temperature that you are choosing, but the order of magnitude is the same. Another important thing that one has to have in mind when you are doing this type of analysis is that when you're heating up your protein, you're unfolding your protein, and unfolding is not usually a reversible phenomenon. So some cases, like the one I'm presenting here for a protein that it's actually big, is not the rule of thumb that small proteins will be folded reversibly and bigger. No, it depends on each particular folding domain. Here we have a protein that you can see that the unfolding and the refolding is reversible, while this protein that it's actually small, it's completely unreversible. There are different models for calculating KDs, considering reversibility and also considering non-reversibility. So again, you have to choose properly the model. And yeah, that is everything I wanted to say. If you have any doubt, and please start using our server for doing the quality control of the data that you are collecting. Thank you.
I hope you caught the applause. Has anybody got any questions for Maria? You'll have to wait for the microphone, I'm sure. So whilst the microphone's getting there, I noticed that some of the examples you gave of the membrane proteins were oligomeric themselves. And I wondered if you could correlate the thermofluor analysis with that oligomeric state. You seem to choose photometry as a preferred technique, but can you get any information from the thermofluor?
Yeah, exactly. It's a very good question. So when you are having oligomeric states of complexes, when you try to do differential scanning fluorimetry, for example, you have so many reporters, so many different domains that start to unfold that the signal becomes very messy. You don't get like one curve, one nice transition. If you are lucky enough and the unfolding of those domains have different TMs, then you can separate, and sometimes you can see like different steps in unfolding. But usually it's not the case. You get like a global combination of all those tryptophans reporting, and it's not a good technique. Much more sensitive for cases where you have oligomeric states is dynamic light scattering, to just follow aggregation, or the mass photometry, because it tells you like on the spot how many more or less complexes are present.
Thanks.
Hi, Maria. This is Inês Justo. Thank you for your talk. I think you make a very good point that without a good sample, we cannot really achieve a very good data set, regardless of the methods and how optimized the beamline is and all that. And going to the point of batch-to-batch variation, since that is one of the most likely causes of irreproducibility in crystallography, have you had the case where you have already some QC on different batches, so you already ran 1 or 2 different analyses, but still the crystallization process is not the same between the two? And if so, what would you recommend as an approach there?
Yes. So it happens that sometimes you get crystals with one batch and you don't get crystals with the next batch, and then it happens a couple of batches and then you get crystals again. So something is going on. There are three things that one has to do, like like let's say the fingerprint of your protein. You make a simple CD spectra to see that at least the secondary structure is conserved. If you can run a thermal shift assay to know that the TM has not changed, so you more or less expect that the TM isn't changed, and dynamic light scattering ideally to see that you have no aggregation. So with those three things, you can characterize your sample and compare to the previous batch. Still, it could be that, for example, you're having some oxidation, and this will not be reported. If the oxidation is not translated into a secondary structure difference or an aggregation problem, then you don't see it, and then you cannot crystallize your sample. So at least make all the checkpoints that you can do. Sometimes it still happens that it looks good and you still cannot crystallize. And there are other processes like oxidations or isomerization of prolines, for example, sometimes also.
So I have one question on Slack from Jason Guo. How accurate is mass photometry in terms of measuring the molecular weight? So if you have a 10 kilodalton binder, would you be able to see the difference?
No, it is a technology that is not giving you the exact mass of your protein or complex. For that, you need native mass spectrometry. Actually, mass photometry cannot detect things that are smaller than 50 kilodalton or 30 kilodalton, depending on the generation of the device that you're using. The one that we have is not the oldest one, so we cannot detect things smaller than 50 kilodalton. The new ones claim that you can go until 30 kilodalton. So if you are working with big complexes, it's super useful. If you want to see the binding of two proteins that are 25 kilodalton, you will not see it.
Okay, thanks. There's another comment from Leonel Costanero about the use of wavelengths in UV spectra. Perhaps you could answer that online if you have a second on Slack, which says, I always measure up to 400 nanometers, not 340, as 340 actually indicates aggregation. So. Yeah,
you can see the scattering later. Also, it depends of the protein. Some proteins that you are using, you know that they have chromophores or so you need to look at different wavelengths. It depends on your system. But if you are measuring or checking something that binds to nucleic acids, for example, this is something that you have to do.
Okay, thanks again for your talk.